Hi, this is Matt Montgomery, Burr Sales Agronomist. Some exciting technology may be coming on the market in the next 15 years. That exciting technology relates to rootworm management. It goes by the name of RNAi. Today, let's paint a rough picture of how this technology will work. Hang in there, though, because describing how this technology will work requires lots of background material. So let's start by reminding ourselves why new rootworm management technology is important. I've posted the four, what you would call pillars of corn rootworm management on this slide. These are the BT-oriented tools that we have to date. This was the first rootworm BT trait on the market, and this tool will be the most recent addition before very long. They're all great tools, nice things to have in our rootworm management arsenal. However, there's a really big problem developing. The first of the rootworm management traits seems to be encountering resistant rootworms in some portions of Illinois and Iowa. In addition to that, researchers have been able to induce resistance development to every currently available trait in the laboratory to make it happen artificially. The point is this: resistance has developed to a portion of the trait spectrum, and other traits in the spectrum, when used long enough, will likely exhibit resistance development as well. So where does that leave us? Well, it reiterates the need to rotate traits to practice resistance management, and as we rotate traits, it might be nice to have another tool or two to rotate to. So what's next? If we need to rotate tools to keep as many rootworm management tools viable as possible, what new tool might be coming on the market? That's where we enter with RNAi. RNAi is a prospective and promising new technology for rootworm management. While it's a potential rootworm management tool, it is very different from the traits, the BT traits that we use for rootworm management that are currently on the market. It's not a BT trait. RNAi stands for RNA interference. So what is RNAi then? Well, to answer that, we really need to remind ourselves what RNA is all about. As we go through these slides, just remind yourself that when we talk about BT, we're talking about basically that protein forming ulcers, wounds in the gut of the insect. This is very different. Let's take a moment to look at an animal cell. Remember, there are all sorts of different parts to a cell, all sorts of different what we would call organelles. Now, one of the most important organelles is the nucleus. The nucleus contains the genetic material of that cell, the DNA. DNA provides instructions about what needs to be going on inside that cell, what needs to be produced to keep the cell alive. Think of the DNA as the board of directors, the instruction provider within the cell. Now, it's one thing to know what needs to be made and to say what needs to be made, but once you figure that out, you have to actually make the stuff. It's fine to know what needs to happen to keep the cell alive, but if the cell wants to stay alive, it better actually get the stuff done. A board of directors doesn't do that. A factory floor does. Enter the ribosomes. These are the protein factories within the cell. They make the stuff that's needed to keep the cell alive. You need both, right? You need to know what needs to be made to sustain life, and you need to actually make it. You also need something else, but we'll come back to that in a bit. Now, it's probably been a while since you studied cellular biology, so humor me a bit here and let me remind you what DNA looks like. It looks like a ladder. But that ladder has a little twist to it. Just think of a twisted ladder and you have the idea of what DNA looks like. Again, this twisted ladder is the board of directors that tells the factory floor what to make. Now remember that I said we also need something else in this little community. Well, if we only left it at the board of directors and the factory floor, we'd have a problem, a very big problem. This is the big problem. We have no plan for how you get instructions from the board of directors to the factory floor. There's no phone line, no internet connection, or texting available between the two. So how do we get information from here, the board of directors, to the factory floor? How do we get that important stuff made? What's the solution? Well, we need a messenger. We need the information about what needs to be made to be put on a disk, and we need that disk, that messenger, taken to and read at the factory floor. Now we've finally gotten to RNA, messenger RNA in particular. Now RNA looks like this, a half ladder. Remember, DNA was a full ladder? What is important to remember for today is that RNA is the extra player we need, the player we must have. RNA is the disk that the board of directors puts its instructions on that the factory floor reads to start production. So again, here's the pathway. The board of directors, the DNA, says this is what is needed to stay alive. The instructions are put on a disk, messenger RNA. The disk is then presented to the ribosomes, the factory floor, which makes what's needed to live and all is well. Board of directors providing the instructions put on a disk taken to the factory reading disk, and beginning production as long as you have those steps you live. Mess up any part of that community and you die. So let's venture back to RNAi, RNA interference, and let's talk about what that is. Rootworm RNAi is thought to use the rootworm's viral defense mechanisms to kill rootworms. Well, that's all fine and dandy, but how exactly does it do that? Well, more background materials needed, so let's briefly talk viruses. In the upper left corner, we've depicted what viruses sometimes look like, half a ladder, kind of like RNA. Again, this is all important discussing how RNAi works. For a brief period of time, a virus will look like this. It'll look like a full ladder, and if left to its own devices, the virus will trick the cell into making copies of the virus instead of the stuff it needs to. I guess it's kind of like the virus is a pirate and it's taking over the ship and doing what it wants with it. And what that virus does is make lots of copies of itself. If the cell's factories are busy making viruses, the cell's factories are not making the stuff needed to live. Remember, we needed the board of directors, their instructions on a disk, that disk taken to the factory reading the instructions, and then making what stuff was needed to live. A virus wrecks the back end of all that. Instructions can be taken to the factory floor, but the virus has overwhelmed the factory with its demands instead of the board of directors' demands. Well, anyway, the cell can't let that happen. So when the virus changes into a full ladder, the cell says, hey, something isn't right here. I don't think that's supposed to be here. The cell looks, it says, you know what? I don't think that double ladder is with the board of directors, and it decides to do something about it. So the cell then releases a little molecule that attacks the virus. That little molecule acts like a pair of scissors. Now the ingenious part about this is that some of the little clips of that stuff that was chopped up, some of those little clips that remain, are then used to combat the rest of the viruses in the cell. As those little viruses, those half-ladder strands float around in the cell attaches one of these little clip pieces to it. Remember what happened when the virus was a full ladder earlier? The cell chopped it up, right? Well, something similar happens when the cell notices this partially full ladder. As a result, the virus gets chopped into pieces and the cell is saved from the virus. In some ways, I almost feel like I have to apologize because we've gone through an awful lot of stuff. We've talked about DNA, we've talked about RNA, ribosomes, and viruses. We finally have enough background to describe how RNAi works. Remember our little community from earlier? You have to have the disk, RNA, carrying messages to keep the cell alive, right? Do you remember what this looks like? Remember those little pieces of the ladder, those little pieces of the virus, the cell attached to the virus? It chopped up those little pieces and then stuck the little pieces of the virus onto the other side. Remember what happened when the cell didn't like the look of the thing and chopped up that partially full ladder? Well, that's what we do with RNAi. The corn plant will produce little pieces of the ladder inside itself, and those little pieces of the ladder are designed to attach not to a virus, but to the cell's own RNA, the cell's own messenger disk, the thing carrying the important information. So the rootworm larva eats some of the root material with these little particles, these little ladder pieces in it. Those little ladder pieces are then attached to the RNA, to the messenger disk. When that happens, the viral defense mechanism kind of kicks into gear. The rootworm cell is confused because the rootworm ate RNAi plant material. It's now treating its own RNA kind of like it does a virus, and it's chopping its own RNA up almost as if it were just a virus. So now the rootworm doesn't have all the stuff it needs to live. There's no longer a disk with information on it that tells the factory what needs to be made to keep the cell alive. So the DNA has all this information about what needs to happen, but the information isn't getting anywhere. That spells death for the cell. And eventually, when this happens a gazillion times over, death for the rootworm. And it's all because the RNAi corn plant produced little pieces of ladder that trick the cell into killing, or in some cases, silencing, its own RNA, its own messenger disk. Well, this technology is still a little ways out. We're talking about 2017 to 2025 before we probably see it on the market. Now, while that all sounds neat, we all naturally wonder if it will really work or if it has been shown to even work. And actually, yes, we do have documentation that it can work. Nematodes, for instance, have been fed those little particles in the lab and have been killed by it. And as far as resistance risk, who knows? Now, some fungicides, like Apron and Allegiance, don't use RNAi, but they do shut down RNA. And we know that resistance can develop to products in that fungicide mode of action family. Additionally, there seems to be a little research that demonstrates how RNAi resistance development could work. While the traits will be pyramided with other traits, it won't just be a standalone product, those points that I just talked about related to resistance development reiterate what we have talked about earlier. Even this new technology will require resistance management if we want it to be useful over the long term. Thanks for letting us provide you a brief glimpse of the future. Stay tuned for more in-depth agronomic information at Burris Agronomy U.